So, let's say you want to grab a cup of hot chocolate. In order to grab that cup of hot chocolate, your muscle will need to contract, and that will make your arm move. So, it all starts with the brain sending a signal down through the spinal cord, out through a nerve, and making its way to a neuron, which is this right here, neuron. The neuron then connects itself to a muscle cell, or as you can see, a muscle fiber in the diagram. This is called sympathetic transmission, also known as neuromuscular junction. Neuromuscular junction is when the transmitters are connected to a neuron and a muscle fiber. When the signal gets to the neuron and connects to the muscle fiber, it is going to travel through the sarcoma, which is this right here, and down the transversal T-tubes, which is this right here. The transversal T-tubes are deep inside the muscle fiber. The transversal T-tubes are then going to interact with the sacroplastic rectum, which is this right here. Now, since the signal interacted with the sarcoplasmic, it has now created calcium ions, which are these little circles I drew. In the presence of calcium ions, the myosin and actin, the thick filament, and the thin filament, are going to want to contract, what makes the muscle move. This is also called the sarcomere. The sarcomere is underneath the sarcoplastic rectum. So this is right underneath this. So now the myosin and actin are going to contract due to the calcium ions, which is gonna make the zigzag line, the Z line, want to shorten. Kind of like this. Pretend my two fingers are zigzag lines, or the Z lines. So they're uncontracting right now, and then they're contracting. Not contracting, contracting. Not contracting, contracting. That's sort of gonna look something like that with the zigzag line. Now let's go to the filament side of the theory. In stage 1, we see the actin, the thin filament, and myosin, the thick filament. In actin, we see the tripomyosin, which is this black line covering the binding sites. In tropin, which is right here. In the myosin, the binding sites are not covered. The binding sites are the myosin wants to grab onto an actin molecule, but it can't because the binding sites are covered, which is going to be a dilemma. Since the binding sites are covered, the muscle now is off, aka muscle is relaxed. Well, in stage 2, let's pretend calcium ions from the sarcoplastic rectum binds with tropin, which pulls the trimyosin off the binding sites. See right here? The tripomyosin is not on the binding sites. This is crucial because now the myosin, which is this, is going to be in a cocked position, ready to grab onto an acting molecule. That's the second stage. Now in the third stage, the myosin head grabs onto an actin molecule, which forms a cross bridge. In stage 4, we see the head muscle, the myosin muscle, pulling on the actin molecule, which is called the power stroke. Okay, let's recap now. In stage 2, the calcium ions came from the sacroplastic rectum and pulls the triomyosin off the binding sites, which makes the muscles ready to contract. Then in stage 3, the myosin heads grab onto the actin molecule, forming a cross bridge. Now, the myosin molecules are pulling on the actin molecule. This also forms a contracting. Now, in stage 5, since we pulled, we need to reset so we can pull again. Relax the muscle if we're not going to contract it anymore. In this stage, we're going to release and reset. Now, this is where ATP comes in, which breaks down into ADP. ATP is used in this process as well as breaking the cross bridge and using up the energy to reset the myosin head back to where it was. See, the myosin head is no longer grabbing onto an actin molecule. So we call that release and reset. Now, we have to acknowledge that if the calcium is still present, the cycle is going to keep on repeating from stages 3 to 5, where it is grabbing onto the molecule, pulling on the molecule, and resetting. Then it'll repeat again if calcium is still present, which is grab onto the molecule, pull the molecule, aka power stroke, and then release and reset. However, if the calcium is not present, it is going to come back into the off muscle, which is relaxed. See how when calcium is not present, the tropin and the triomyosin are right back at the blocking the binding sites, which is not allowing the myosin heads to grab onto the binding sites and pull. And that is sliding and filament theory. Thank you.